السلام عليكم ورحمة الله وبركاته. ورب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. السلام عليكم ورحمة الله وبركاته. Inshallah ta'ala, we're going to be concluding our golden lab for this term, inshallah ta'ala, for BIOC17, class 1, week 10. Inshallah ta'ala, we look forward to having a review, but inshallah ta'ala, for the in-person component, we'll discuss a few things in due time. May Allah subhanahu wa ta'ala accept our efforts in this life and in His remembrance to our hearts and souls find rest. Inshallah ta'ala, look forward to seeing you soon. I'll never know when you're she's on. It's not. Because then we said, no, we can't say that do like three and one if we don't have it. So yeah. Yeah. You look at Mark. Mine's 2%.
I have a method. Didn't put my name on. Did you? Probably. How many times? Yes. Here's my grace. The one that I was doing last night. I was like pulling up the log reading rules on my laptop. I was like, wait, we're back to high school. What am I doing? Oh, that makes sense. You're just used to it. Oh, so you're just, oh, I guess it all. Yes. That's my. That's where. It's not cute. So, yeah, yeah. It's tiny though, you know? Did you do? No, I'm not. Oh, you're still. You C1. You're D1. You're D1. Okay, I'm D1. Okay. I know UTSC. Yeah, I have a D2. It's your choice. D1. Oh. What are you doing with these plates? I mean, the no. Break out and no, I didn't. I did the whole thing. I did the whole assignment, and then I realized the guys like were different from the ones we actually had to do. So I was just having my assignment, and it's like so if I lost when I lost my brother. C9. Yeah, so true. But it was so. Dude, I hate that too. Dude, we never wait. Whatever the hell there is, there's a competition or something. No, it's going to be in the credit scene. Which is like 14 seconds. Do it in one day. And some people might do it. One person did like she did absolutely nothing. It's just enough to your name on that statement. Well, like when she showed up, he was like not and so we was like throw her and throw and I did and she literally was like, I actually can't introduce this. Yeah, so let me know what you guys want me to do. What is I'm not even to the process. Like, it'll come to I'm like, no, and then she literally said, so let me know what you guys want me to do, or like what I should be doing. Watch your work. Like, maybe try to. What's my name? Yeah, she was transferred from UTSC or whatever. But I was like, you know what? We only had each of us. No, like Laka? Yeah. No, what? I guess so. I go, yeah. She just wanted to find those. Yeah, no, super nice. No, I know. Okay. But like she wasn't like you know how on the course out calendar there's no progressive there's like B90. So you don't actually necessarily have to be in any of the classes to do the project. But she went so she did not in any of the classes, so she like genuinely like wasn't for immunology or whatever. So she was just like we're all in immunology and the other girl was in animal behavior and doing it in so we all have something. Well, no, you didn't have. You know, you didn't have that. Yeah. We're all to relate to the possible. Exactly. That's exactly. I thought they were named that. Well, no, you do want to do well. But it's not like it doesn't have to be like. Well, it depends on what we get. You just said you get it. We get it. Yeah. Every announcement that they were trying to bring back to us more. I think because you picked the date to apply on the 11th. So that's fine. Someone's going on the and one other drug.
Oh.
Together. Oh, that's really. She's. She didn't know. No, I did that out of on purpose because I'm a visual and if she moves anything.
Oh, yeah.
It's just OCD. I know it's a problem. That's literally why I had to put it in her. But I'm not kidding, because I was a director. I was a finance fucking director. You know how fucking like stressful that was? I literally couldn't delegate my time. Because if they weren't doing it the way that I did it, or I just wasn't aware, I would have freaked. But I was like, dude, I can't do this. Sorry, I'm not figuring it out. I'm a teenager. Oh no, I know that. It's specific. It's just with people. Like it's like when someone gives me a task, yeah, when someone gives me a task to do, like I'm gonna do that task perfectly fine. But like when I have a bed. I think maybe something was very respectful situation where you have the because no, because there's expectations, like when I heard this one, I'm like, I need to be perfect because like I need to, yeah, exactly. If we got a bad grade, it was really my fault. Other section, it's the it was okay. I'm actually gonna explore my like the video itself is entertaining. It sounds good. The information is that's right. The video. Yeah, I tried to send her the link. Like yeah, that's beginning of like the
So I didn't say this earlier, but sorry. Hi everyone. I hope you are enjoying week 10. This is our last lab, our last official lab. I'll talk about a few things in due time. And then I have your generation time assignments marked down at that end. If you haven't grabbed them, you can, as well as your unknown assignment. There's quite a bit that needs to be discussed today. I'm not sure if you guys were given access to the slides. No? Okay, so I'll just try to go over the things that are shown here, but I also have some external notes that I want to provide to help you guys excel. So just give me a moment. Zihan, when you're ready, you can show me your lab note. Perfect. So in the meantime, I just want to make sure everyone has grabbed their unknown assignment and generation time assignment. Are there any questions regarding the UV FLR that I can answer as I make notes, if that's okay? Let me just see. And yes, apologies as well for the this thing yesterday, the grade confusion. Pretty much, I have a block on Quirkus when I try to adjust grades, and then essentially it went to everyone else's grades. So that's where things got a bit confusing. but alhamdulillah, things got edited back to where they are supposed to. And as I mentioned, pretty much the scaling or like the different things that I will describe to you will be done likely via email, via the quickest messaging system. So thereafter, I'll talk about things that I may be missing for today. Warren, you had a question. Or actually, Eunice, did you have a question as well or no? No? Okay. Let's pay attention, folks. Warren, what's your question?
Do we put the picture or do we just put the table that represents the picture?
Okay, so great question. For your results, you should have two pieces of results. That includes, as mentioned on Quercus, technically three, your observations, right, in paragraph description. You have as well, and that could be separated, right? Because I noticed for some of you, you may have like essentially your tables and figures, like your plates, for example, all clustered in one region. But you want to have it like a storyline, like a magazine or a newsletter, where you have, a caption that's in our case detailed, right, with the species, the different conditions, your title accordingly, and then the brief results that you have in that caption, and keep that in mind that as your figures, they are normally underneath your captions, whereas tables usually have their captions above. And so I want you to have either a table included as well. So you have a figure, right? The class data that I sent on Quercus for Practical 8. And you guys will use as well, like include a table or a graph, either way works, essentially what you see from page 100 of the lab manual. In the lab manual, it talks about essentially the plates. Sorry, maybe hard to see, but briefly, you have your plates, right? Half was mass, half wasn't. Let's say this was it. And this essentially is the mass, especially technically the growth of bacteria. So you want to be able to compare as a metric all of the ones that are on this portion, right? In terms of their turbidity or colonial growth. And there's a scale from 0 to 3 mentioned in the manual. So the idea is that for across the different plates, you want to have a table that showcases, across time. Well, I'm just showing it here, almost like a graph, but the idea is that you had it at 0, at 5 minutes, 10 minutes, 15, 20, and then 25. And then across the different bacteria, and then yes, that would be the case across bacteria. Piece of Phyllis. Make sure, please, you guys, place all details required. 7 days, 24 hours, right? Dr. Bart wanted me to let you guys know that, just to clarify this, you're presenting all data. Okay, so just bear that in mind. And then your values could be a 0 out of 3, it could be 0, 1, 2, 3, etc. Right? Or in our case, likely, if you follow the hypotheses and your predictions, it likely would actually decrease over time, right? So this made me think about like 2, 1, 1, 0, 0, etc. And so those different values and the increments accordingly would likely change. But you want to once again have a detailed caption above for tables and below for figures. Does that answer your question, by the way, Mary? Okay, any questions regarding the FLR as well? Yes. Oh yeah, so I forgot you guys don't, well, you don't have the plates themselves, and that's okay. But what you can describe is the things that me and Dr. Brent mentioned the last week, 2 weeks ago, talking about the plates. So there are common things, right? Contamination. The L rods essentially not like being burnt or like being too hot to essentially spread your bacteria. There is as well, misuse or essentially using the wrong bacteria in your specific plate, right? So you hypothetically could have switched the two time points for B subtilis. There's so that's mischaracterization. You have aseptic technique for cross-contamination. You have the volumes, right? Because you would have used 0.1 mils, but if you accordingly use other ones, then it could influence the progression of your bacterial growth over UV exposure. And then the other subtle point, which is probably a big one, is essentially having the lids on versus off. So those are 5 main ones. I'll repeat it in case anyone wants to take note. You can at least mention 3 of them and describe how you would have resolved those. So actually discuss the significance of those limitations and what you would have done to resolve them. Sometimes you may say like, oh, these are my limitations, and then call it a day. But you need to describe them in depth, so that way I know that you guys are critical thinkers. So bear that in mind. You have cross-contamination for aseptic technique. You have mischaracterization, putting the wrong bacteria in the wrong plate, or like mislabeling your plates. So that's second one. The third one is of, I said aseptic technique already, volumes, right? So measuring or placing the appropriate amount of bacteria into your plates. The fourth is your L rod, right? Because if it's too hot, then and essentially if you had direct presence of 70% ethanol, you're likely already killing your bacteria, right? So that would overestimate UV's exposure to bacterial growth in the different treatments that you would have had. And then the fifth one was the impact of the lids, the plastic lids on your essentially apparatus or on your results for UV exposure. So you could talk about 3 out of the 5 or as many as you can within that within your page limit. Let's see. Any other questions? And if you think of others, of course, you can describe them as well. Like try to think critically, describe limitations, extensions. I'm looking for these details, right? And each of them have an according mark, so you want to bear those details. Yes, you can. You can. Great question. You can as well. Any other questions? I know some of you also have already uploaded yours, so if you want to actually make updates based on what I've said thus far, you can, and that's not a problem. Any other questions? Questions? Let me just see something. Yes? Can you repeat it one more time? So you want to also include paragraph description. And in fact, what I recommend is having them in 3 pieces, reflecting a brief summary, right, for your results, talking about what your aim was at the beginning and what you did to essentially explore that aim. And then, like, which is analyzing the effects of UV exposure on different types of bacteria. You could specify those bacteria as well. But nonetheless, you talk about that. That's like your intro to the results section. That's your paragraph description. Then you're referring in that paragraph description to your first result. It could be figure 1 or table 1 or graph 1. But essentially your figure 1, which is likely going to be described then, like next. And then have a caption, once again, describing what you had in detail in your figure. Thereafter, the second portion of your results, you're going to describe actually what you see from your plates, right? So that's where you're describing your plates. You referred to it at the beginning, but now you're going to describe them right after you've shown your results for the figure. Once you're done that and describing that in depth, you can go to your table, refer to your table in your second portion, and then thereafter, inshallah, conclude with what you have from your results from the table as well. Does that make sense?
Picture, is that not or like should the caption be like a less detailed version of that?
Technically, captions should be a bit detailed, like in the sense of like what those conditions are, because regarding the captions, They mainly show your methods, like if you have a methodological approach, plus a brief summary of the results. Like it doesn't have to go through every single bacteria across every time point. It's just very brief. Whereas for your and I didn't write down a brief summary of results there because maybe hard to see, but for your paragraph descriptions, that's where you're more elaborate. That's where for your paragraph descriptions, you're more elaborate there. So you're describing more in detail. This is what I've seen across, E. coli versus Staphylococcus epidermidis versus the two different types of B. subtilis. So you're more elaborate there, whereas in your captions, you're generally you're introducing, you have a title, right, for your captions, but then thereafter you briefly touch upon your methods or the conditions that you have, be it in your figure or on your table. And then at the end, you're describing briefly your results, pretty much your major takeaways or the trends that you've seen from your results. Yes.
So,
Yes. Yes, at the bottom. Yes. So the same trend occurs for figures and tables. It's just the orientation, the order, / arrangement. Tables have captions above, and then figures have captions below. Any other questions? No questions? Yes. The quantitative, essentially. You could. That both ways work. That's sound.
I think you said that you wanted the written results, but if you want the bigger than be like incorporated into the result, but if I do it, like, it's like if I write like one paragraph for each of the cultures, then both the figure and the table are going to be on. Like, if I first figure out, I won't be able to put the writing in the figures.
So, okay, that's a good point. Pretty much to make it simpler, have the intro as we talked about within your results, and then your second paragraph would likely be qualitative based on what you've observed from the plates themselves. So describe them across time points, describe them across bacteria, pretty much what I've said on purpose. And then for your last table or your last result and last portion of the results section, you're describing more in depth the quantitative differences across bacterial growth following every exposure time points. Does that make sense? Yeah. Okay. Now let me just go. And if there's other questions, if I have time, I'll try to answer them before 10 p.m. today. But you just want to bear in mind that you have pretty much what you need on purpose, my messages, the tips that I shared with other people. So I hope that will be a benefit. Now let me go through your code. And there's a reason I'm talking about this now, because technically you will have 2 more weeks, including this week, to boost your grade if needed. And what I mean by that is, so I try to make sure that you guys have the material that you need to excel. So next week, I'm planning to do a review. If you've probably seen from my Quirkus message, we're having a lab review pretty much at the same times that we'd have this practical. The times are essentially from 10 to 1 for Tuesday and Thursday, and they're going to split up the lab reviews into labs 1 to 5 for next Tuesday from 10 to 1, 10 a.m. to 1 p.m. inshallah. And then for labs 6 to 10, including this week, we'd have that review for Thursday, so next Thursday. So bear that in mind. And I'm going to talk about this now, so that way you guys can prepare. And if there's things that you want to change or improve, this is your time to do so. So I've given you digits, letters, alphanumeric codes, and essentially, let me just give you some context. Okay. Maybe my last marker. So essentially, let me describe what you're seeing.
Wait, sorry, the review for labs 6 to today is on Thursday next week
Yes.
from 10 to 1.
10 to 1.
Is it recorded?
I can, but I would recommend you guys coming because.
Yeah.
Oh, really?
Yeah.
So maybe I should adjust it. What exactly, or what's the time?
From 2 to 5, but I think you can understand when we say that we want. But it's your. I have stats on the stats exam at 2.
Okay, so nonetheless, we'll talk about this. If it impacts everyone and even for yourselves, we'll try to see what we can do to make it needed. Let me just actually talk about those codes. Let's see, I think I have other things actually. So the grades, you have your grades there. Let me just see. I hope everyone. Amman, you have yours, Rain, you have yours, Misha, Ishma. You guys can come here and grab your graph. And this leads me to my next point. You guys actually did an amazing, superb job. And what I mean is, so by the way, unknown and generation time assignments are there. The breakdown, I believe, is also provided. For the generation time assignment, I'm actually very happy. You guys did an amazing job. The average was an 87, so that's really impressive. I would say, like, in terms of takeaways, always follow what I've provided, right? Be specific with the bacteria, the increments, the different components I've mentioned. And I've scaled the grade to make it a 90 as an average, but the caveat is I may not be able to scale your final FLR. So just bear that in mind because technically speaking, there has been a bit of scaling on my end, and I don't want to make it unfair, essentially, because now that you have all the material that you should have for this UVFLR, you really have to make sure that you've put in the effort and the time that I've suggested from before. So just bear that in mind. If you're missing anything, just let me know as well. Any questions before I talk about codes? Any questions? No questions? Okay, let me talk about the codes. C1, so I have them in increments actually. I have to make an edit there. So I don't want you guys to get confused, so let me just go over this. Okay. I want you guys to see these codes or understand where I'm going with this. Hopefully you have them written down. So C1 is an 80 to an 85. C2 is an 85 or an 86 to a 90. D1 is a 90. I don't say 91. Okay, we'll say 90 to a 95. And then a D2 is a 96 to 100. This reflects your performance in the lab. Normally, your lab notebooks are based on completion. I would say, though, some of this is incorporated with what I've suggested and what I've described from before. And so what I want you to do, two things. One is I want you to think where you would have stood in that range, right? Because technically they're 5% increments. So I want you to think, imagine to think more ambitiously, you're on the lower end. So that way, during these next 2 weeks, potentially, you can upscale your grade. And the goal is that you guys could be able to hopefully try to boost your grade accordingly from one level up to another, hopefully. I can't make promises, but I just want to give you guys this heads up now so that way you guys can compare if there's any outstanding things that you need to complete. And once again, performance is modulated by one, the performance in the lab, two, proficiency, like being able to work with your group, try to complete tasks as effectively as possible. And three is participation, right? So sometimes I ask questions to you guys, and my goal is for next week to, inshallah, give you guys more opportunities to participate in the lab. So try to be as reflective, as integrative, as participatory, or participate as much as you can. So that way I could accordingly evaluate you to what you rightfully deserve. So, and bear that in mind. I think Stephanie can attest, for the different courses at People TA, there's certain guidelines, right, that students have to excel, right? And I try to incorporate that level of justice through the rubrics and stuff, but also to provide you guys the opportunity to get scaled up accordingly. If there's any room for my own improvement. But the task for you guys is to also improve. So see for yourselves if there's a way for you to improve based on what I've said on Quirkus via emails and other ways as well in person. Any questions? Any questions? Increments? Okay, pretty much. I feel like I don't have time to actually. So let me, because we're a bit tight on time. I'll talk about them. I'll talk about it in due time. And let me just go briefly to other material. Let me see. Lysozyme assay. Okay, so we have the material that we'll need. Lysozyme assay will actually do that because we have, I placed on your benches your plates from last week, MSA, Mannitol Salt Agar, McConkey, and DNase / hydrolysis test plates. And then these are your gelatinase test tubes. So these are your tubes accordingly. You can see this one's solid, right? There means that whereas this one is liquid, right? So you guys can probably tell positive versus negative results. And that's where this is the control, but hopefully you guys will be able to interpret your data accordingly. This is in prep for the other plates. Yeah, so you have your plates. I want you to observe them as well. The lysozyme assay, I want us to take care of after, so for maybe in due time. Because I want to make sure that you guys have the material removed for other things. The counters are here as well on your bacterial growth. Generally speaking, what you need to do for this week is, because it's our last in-person lab, you want to be able to actually do your post lab this week, meaning you want to actually draw out your figures, right, with the captions for the different plates that we have. For your results for a gelatinase test, you can actually just describe them. You can make a table, but describing them should suffice. And then, so that's your 4 exercises from last week. For this week, sorry, there was another. So we had the Snyder test, blood agar. We have the biofilms, but there's one other thing. I think I'm missing one other thing. Which one? Yes, those 3 plates right there. So you want to record the results from those. So I think one more. TDT, yes. So TDT result, we're going to go over a bit as well from the microplate reader. I just want to also give an update. I did make an update to the file, right, for those that weren't able to see on Quirkus, because technically from 0 to 5 minutes, your bacterial growth should increase or decrease? Increase? From 0 to 5 minutes. Decrease? I'm taking note of this for participation as well. So decrease or increase? Decrease. Okay, so decrease, right? So bear that in mind. So when you look back on Quirkus, if your results were like this, like an X shape for like 50 degrees and 65 degrees, you should actually make the adjustments such that for you guys, it looks like it's decreasing over time. Just bear that in mind. You can make that correction as well today. But briefly now, you have your plates, you have the gelatinase test tubes. You can actually get started with observing them now. And that's pretty much the results from slide 5. I'll talk about TDT after 10 or 15 minutes, but I want you guys to look at those results for now. And then actually for the next 10 minutes, try to do that. And then I'll walk you through your biofilm assay. Any questions? If you have already observed and made notes of your plates in your lab notebook, that's amazing. But I want to give you guys maybe 9-ish minutes until 10 to be able to describe or make note of those details. If you are curious regarding your lab notebook grade, because that's a separate grade, then let me know, like if there's any outstanding items. Some of you do have some that I want you to follow up with me, so just let me know. I mentioned to Angela, for Eunice, I think I have one for week 6. So if you could show it to me, just to confirm. Daniela as well, week 6 material. Okay, it's mainly week 6 actually. So let me just go over names. Don't take it like wrong. I'm mentioning your name because I may have to just validate what I have here. Joseph, Misha, Tasnuva, actually, sorry, not Tasnuva, Krista, Aman, Angela, Eunice, and Daniela. Those are the names of people I just need to validate. Week 6 material, and then week 8, if Vishwa can just confirm post-lab material, that'd be amazing. So in the meantime, your other group members can just make note of the details, what they've drawn out in the lab notebook, and then that should suffice for their completion. And then when you guys come up, you can always return back to see the other results. But just try to show them to me. As well as Aman, show me your lab notebook for this week as well. Okay. Yes, yes. Yes. Forgive me, Anisha? No, sorry. No. I have to say it. As you have, sorry. We told it. So you don't have the results, only have for your group, so you don't have it for other groups. Okay. Super. And I have anything to see from you? Can you see the last page? The goals. Is that yours? Okay. Perfect. Thanks, man.
It was the kind of inside. It's
Okay, last. This is
and with this
week 6.
sorry. Okay, here's the results. The critical discussion.
So results you need to have for that.
Okay.
So if you can, right? And then you can still say anything.
Then to check the pull-up word. Lessons.
Yes. Oops. So I think it's a post lab actually.
No, this is a lab.
Yes. I need your post lab actually.
Oh, okay.
Let me just make sure I take notes. Actually. Oh, actually, you're going to show it to me. So show it to me when you're ready. The edits.
Lab 8? Here? I think I just showed you.
Technically, it's week 7 because it's a post lab. You know like when you come into meetings. And so presentation.
Here's the results. Big discussion.
This is good.
Okay,
So I'll just make the check right now.
Okay.
I just want you to go show me the plates with the thank you captions, and then you show me that.
Okay.
Perfect. No problem. Who is first? I don't. Yeah. Okay. You know, the reason I'm doing this is so hopefully all of you guys can get 100, right? So, and by the way, folks, like not every TA will do this. They will literally just mark and grade you accordingly, right? So I want to make sure that I could, attest to your performance and your lab notebook quality. Sorry. Of course, yes. Thanks. I appreciate it. Thank you for Sorry, can you go back to two? If you can, I can't see it. Okay, can you go? You can continue. And do you have the results for the second test? Because I know they're in like a description. The second. The catalase and hydrolysis. I see, actually, I see it for there, the beta lactase. But do you have one for catalase?
Yeah.
I think, sorry, or
Assignments
okay,
and
you go.
projects.
Can I just confirm? Permission? Permission? Thank you.
Like you did all that stuff. Period.
Things like that, because what we're doing right,
What
what
was
I'm
the
doing right now. For my post lab for last week is the McConkey test, the Jazakumullahu test.
Yeah, yeah. But I don't think it's not for this week. It's for
You did
week 8.
like with your own voice. Oh, that's cool.
The one that I just
We did not.
sorry, week 8 mean post lab.
The tens is this.
I'm excited to watch.
We had discussed mine, like.
And you include the figure
Yeah.
calculations for
What?
the
No, by heart. Like that was a
Perhaps
specific.
then I didn't catch you, so perfect. Thanks for confirming. No problem. Count. So this was if you go and you just want to
Yeah, exactly. No, in, it says like.
yes.
But without cutting out like the breathing. It's like.
The breathing.
Yes, I remember. Oh, did you calculate it?
Yeah.
Okay, and these are the things.
30 seconds of breathing.
What are these ones? So this is transmute.
That was initial time.
This is for the generation time side.
Yeah.
Okay, okay.
Yeah.
I see.
Yeah.
Can you just write that down? Huh?
Because,
But it's still important. So that means like it would be. Okay. I think that's perfect. So no problem. Yeah, that should be good. One more page just in case. You go one.
Oh, yeah, man.
So that's
That's the intro.
yeah, one. Okay. Let me just want to make sure I give you.
That's just.
You're great. Perfect. Thanks a lot. I think it would be Krista. I really appreciate it. Give me one moment. Okay.
What was it you wanted to review week 6?
I believe so, yes. Week 6 and 8. Actually, technically post pre-lab 9 and then pre-lab 5. I think post lab. No, just
Oh, just it's
For both.
okay. This
Really pretty.
this is post lab for week 8.
That's
This
okay.
is-
That's okay. So I'll check that. Let me just see. Okay. The other thing I would just recommend, because it seems like the discussion is quite. So I would actually recommend highlighting a bit more, even if it's just highlighting data emphasis really hard. Because there are some things that they don't see in the discussion questions. So I want you to include whatever you have in this section, and then actually do this section. That will, now for weeks for post synopsis.
Like the that like not
This is good. You need a discussion.
okay.
So this would be yours.
Yeah, that's true.
So could you do the discussion for this on the internet? Like, once you're done, but
So discussion for 6 minutes.
yes.
Okay, thank you.
Perfect. Daniela, are you ready? Yes.
Which one did you name?
I believe it is week 6.
Is it pre-lab?
Folks,
The chemical germicide.
no, generation. Just want to confirm, is everyone done making note of the plates in their lab notebook? Yeah, bigger caption. Yep. We'll be starting the biofilm in the next 3 to 4 minutes.
This is the post-lab question stuff.
Okay. Maybe the next page.
And then
I just see
this is the next.
next week. So do you have a formal discussion?
Like discussion, I have like little paragraph parts for each one. Each different exercise.
Could you make note of post-lab discussion on that paper?
On this paper?
Yeah.
For all of it?
Yes.
Okay, so including the stuff that we did in lab and the stuff that we analyzed from the lab.
No, just the one in that. Just
Okay,
the-
so this stuff.
Just to make it easy.
Okay. So all exercise 4.
It should take about 6 minutes.
That's fine.
Yeah,
Okay.
perfect.
Yeah.
Thank you so much. Yes. This two, I believe for you it's week 8.
Yes, so that I got sick on that day.
So give you guys 4 minutes to take note of the plates.
By week 8.
Just see them. So visual flowcharts is from
There is no kind of a visual for this, and that's why there is no visual.
see for the broth and the 2 different organisms you could. Let's just have a look. This is for, so you know what I would do? I would make teams. So 1, 2, 3, 4, and then 5. 5 is your control. But you have all of the other ones with specific bacteria. B-subtilis, E. coli, Staphylococcus epidermidis, Staphylococcus aureus, etc. So I want you to make note of the ones that you see for the different conditions, and then just multiply or like summarize briefly what you observed based on, or sorry, summarize your flow chart based on what you have for those reps.
Okay, I will do it.
Perfect.
And rest, anything else you want to check it, because for the other which I am supposed to have a type with the flowcharts.
Apart from that, it's just a visual for exercise 1 and 2.
Exercise 1 and 2.
Yes, that should be good.
Can I do it somewhere here? So that
Yes,
it will be
yes.
proven?
Thank you, Islam.
Anything else?
That should be. Let me just go quick. That should be it. Did I see your book today, right? The one with
You
the-
did, but you want to double check.
Just in case. I think you read through it. Thanks.
You double check, because you said you put too much information which we are not supposed to.
No. If you put more than needed, yes. So it's okay. This is good. Great job. Okay. So, so far, folks, are we done in terms of making note? Aman as well. I need to see your lab notebook. You can Aman go. Okay, you're included. Okay, yes, sorry. I don't think I have anything for you. Actually, I do. Week 7. Can I just have? If it's sure that me. But don't forget, if you can, try not to forget. Narration.
Well, the questions I answer, like,
I just need
Sorry.
a valid.
oh, okay. So there's like the questions, the questions, and then the. Do you need a figure caption?
I believe yes.
Okay.
You can make a figure caption. And by the way, what code did I do?
What?
C1? You're a D.
Oh, okay.
So I just want to let you.
Okay.
No problem. Take the weeks. Okay. Let me just in case, but you still have it, you know.
I have the results because like we had to the summer.
Can you show that to me?
So yeah, show that to you. But I can send it to you as soon as I get home. Like the feed for today. I don't think it's
What's
crazy
that?
more. Yes, I can send all that. So if you want week 6.
Let me just see if there's anything else. And week 3 material. Yes. So, you can do a little bit even on your
Yeah.
it should be okay. Alicia as well. Alicia? So, folks, let's see, I believe we have everything done thus far.
Yeah.
Yeah, thank you. That's all. So let's go to the TDT results, folks. Try not to open them, by the way. Try not to open. No, try not to open. So, folks, TDT, I'm going to talk about that now.
Okay, but it's not E. coli, okay, so your DNA plate, and it's not E. coli that's causing your increase of cultural display, that's possible.
Okay, folks, so let's pay attention. If anyone has any outstanding items, we'll talk about it at the end. But for your TDT analysis from the microplate reader, I want you to have a look at the slide, or at least on deck, essentially on Quercus, what you would have seen from the microplate reader. I mentioned earlier, the viable counts have been updated. So if you haven't already done them, made the update for 50 degrees, please do so. But just to reiterate this, what you're supposed to be seeing here, this is a bit interesting, but essentially, you're supposed to have 2 different reps of 50 degrees and 65 degrees across time. It seems like the one that would be more reliable is row D for at least 65 degrees. And then what you're seeing in comparison for rows A and B would be 50 degrees as reps. But essentially, they go up in increments from 1 to 2 all the way to 8. And they go up in 0 to, sorry, that's the blank. I didn't mention that. The column 1 is the blank. And then from 2 to 8, you have them going up in increments, starting at 0 to then 3 to then 6, 9, 12, 15, and then 18. And the idea is that compared to the Bible counts, which you only had from 0 to 5 minutes, this would perhaps and ideally give you a more readable read for comparing your values and to determine the TDT and D value of your plates. Oh, sorry, yes, of your plates using semi-log paper or from you've drawn in your mind.
That's why I use it separate.
But for like the actual curve from the slide, it was showing them.
Yeah. but more importantly is, you're seeing a slight increase with 50 degrees, so it's not being killed.
Yeah.
You have a little issue between your replicates of your 65, because you see what the bottom one is doing
Yeah.
versus the other one is showing a slight increase and then it's sort of all over the place a bit. So there's something a bit off. In terms of your cell counts, they were fine. So using that data, they should, and the cell numbers, they should calculate the D value and see where they are. D value should be under 5 minutes. And if you look at everybody else's thermal death time, that's what you're seeing. Something is not amiss in the way in which you're 65, because your 65-degree data, which is you're showing 2 different trends.
Yes. And I actually mentioned that what they should focus on is rho D essentially because of the values. Rho D is what they should focus on. Because essentially the trend is not fluctuating up and down in the
But remember that material, your starting rho D is also very low. It's why you get negative numbers from this. There's never any factor in there. So, but that's fine. You can do that for both sets of data, or you can reverse to a different set like Fraser's data, which is much clearer in terms of 50 versus what you have in reading that.
Okay.
Something's not amazing in the duplicates at 65 degrees.
Okay. I appreciate it. Thank you for letting us know. So this indeed, actually, the good news is as you go down, and so there could have been like within the incubation, because as you probably are aware from the microplate reader, there are certain steps that are made in place operationally. So as you go down, as you could probably appreciate here, at higher time points that go actually to essentially like 4 hours time, you'll actually see the decrease, right? So I don't know if I specified this, but what I would recommend is that you go down to essentially compare these results. At 50 degrees, the first 2 reps at the earlier time points are ideal. And then at for 65 degrees, you'd be looking at the later time points towards 4 hours. And hopefully, at least for column C, it looks, or row C, it looks more promising in this regard. So you could instead just focus, if you want to essentially observe the changes over time to determine TDT and D values, then this would be your mode of navigation. It's a bit hard to draw out, but I want to see if I can draw and show you guys what I mean. So technically from the slides, it does say to actually have it in log. Reason being is because your viable counts. Sorry, she made it. Dr. Rabb made a good point, because for the microplate readers, there are in decimal ranges, but for the viable count, you do it in log. Something to note out though, when you're drawing this out, if you haven't done so for your post labs last week, bear in mind that for the earlier time points, right, you're going to be focusing on 50 degrees to be able to derive the change in absorbance readings or the slope to determine the TDT, which is essentially the time that it takes as you extrapolate your line to reach 0 at the y-axis, essentially for the values to reach, let's say these were your points. Your TDT, let's say for one example, at 50 degrees, essentially was from 0 to 5. You extrapolate to essentially the end of the x-axis to get your TDT. And then the D value, which you do for both 50 and 65 degrees, for the time points where you're just clearing in mind 65 degrees, you're doing at the end for the third row. And then, inshallah, what you're going to do thereafter is determine the D value as the one-fold decrease, meaning that like you're going from 10, let's say 10 to the power of 6, 10 to the power of 5, or 10 to the power of 7 in CFUs to 10 to the power of 6, which is more understandable from the curve itself. So make sure that you followed through with what you've done from the generation time assignment. And if you want to show me those results, you definitely can today. If there's anything outstanding, you can show them to me via email as well. Just bear in mind that I want to make sure that I can grab or get as much information as possible in person so it doesn't get forgotten from yourselves and also for myself. So from me and yourselves included. Any questions for TDT or D values, D reduction values? Yes, so it could be so the benefit of this, I told you guys to do the viable counts, right? Some of you may have done also the microplate reading results. If you haven't done so, just make it very simple. Before or by the end of today's lab to actually just draw out the graph and just try to see what you could actually determine. In theory, it would be under 5 minutes, but even from the results I've seen from you guys, they're more towards maybe 20 minutes, which could have different confounding reasons. And you could describe what I've touched upon for your FLR limitations to describe how they could also relate to the results that we had here. One is that this is an open space, right? Where I was trying to essentially do the inoculation. So it's a bit, especially with, the fluid of people passing through and fro, it's a bit difficult to always keep it controlled, the conditions in a microplate reader. The other issue is that the time frame, right? Because the time in which that it's at 0 minutes here is different from the time that it's 0 minutes there in the microplate reader. So those are two different confounding variables. And then just, also the time frame, because sometimes the increments go off by a minute or so. And so those are things that you may want to also consider as limitations. But you want to bear in mind that these are things that are in practice quite they're replicable, or they're applicable to understanding how you would accordingly resolve other matters in the future. So with that, any other questions or any questions? No? Let me just go back. Okay, so let's see. So I talked about your results for TDT. Let's go to the lysozyme and then we'll conclude with biofilms. So the lysozyme assay is something that is actually quite interesting. Does anyone know a bit about the lysozyme assay of what they've seen from the lab manual? Not from the slide, because everyone can see the slides, right? But what have you seen from the lab manual, or what do you know about this enzyme? Yes, yes. And Jihan. And Ashley. Can you repeat the first part?
I didn't hear it.
Okay. The lysozyme enzyme, the enzyme? Yep. Okay. So great points, folks. Actually, any other points from anyone? From anything outstanding? No. So pretty much, yes, it's an enzyme that is secreted from the mucus lining of different tracts and different organs. You have it in the sweat glands, the tear glands, when you're tearing from your eyes, or perhaps even drooling from your mouth. You have essentially a lysozyme. Probably if you recall, from human physiology or animal physiology courses, essentially it helps to break down certain substrates and biomolecules. And that's where you get chyme, essentially from food substrates breaking down, getting digested and essentially decomposed into chyme. And so that's essentially what you have. And lysozyme breaks down chemical bonds, which is noteworthy for bacteria. And I want you to think about this in terms of what you learned from lecture material, right? Because you do know about the peptoglycan layer, right? It includes peptides and glycans, which usually are saccharides. So the idea is that essentially, depending on your substrate or depending on your bacteria, you would essentially have the breakdown of that bacteria with the enzyme included here. And so even though they're fixed, they're controlled, right? You're controlling for the amount of volumes for both the substrate, i.e. your bacteria, and your enzyme, the lysozyme. You have across time or across replicates or conditions an increase in that concentration of the enzyme. And so as you increase the concentration, the idea is that you would be able to see a decrease in turbidity because you would actually do this on the SPECT20s. And so your substrate, which is your bacteria, would eventually become less and less turbid, as you can imagine. And so that's what you would actually expect to see from the Michaelis-Menten curve, which actually, believe it or not, is related, even though this is a variation, it's related to biochem quite readily. If you recall, like some substrate analysis, if you've taken like BIOC12 or I think it's C12 and C13, I think C12. You may have learned essentially about essentially the effects of substrates on reaction rates. And in a similar manner, if you're reducing an OD, reducing the absorbance, you're reducing the, so you're actually increasing in the effect of that reaction because technically the bacteria is your substrate. So the more, the less turbid, it would be nice to draw this, but maybe I tried to draw that. It's really interesting. Connects you back to biochem. So another sea level. Lysozyme plus substrate, which is your bacteria. You can just say E. coli. Actually, technically, E. coli, you may actually say it's wrong. Does anyone know why? Well, I mean, technically, it does count as a thin one, but I want to be more specific. Let's say, actually, perfect. Name an example of some gram-positive bacteria. That's epidermis. Okay, I want to hear other ones too, maybe. Aseptilis. Okay, any others? And Rosius and Rosius. Okay, maybe one more. Think about last week. Blood agar. Sorry. That's one I think someone mentioned as well. One more. I want to hear one from the blood agar. You can go back. It's okay. I want to hear the species name. Because you actually want to know this bacteria for your lab. Yeah. Lactis is one of the ones from the unknown assignment, which is also good to know though. It's also gram-positive. So for simplicity, gram-positive. But technically negative also works here. Now, following this reaction, you would have the breakdown. I actually want to see if there's another way to interpret this. You do have a buffer, but there's other. Okay, so you would generally have, as a result, OD or turbidity. And I'm just highlighting this now because essentially what you would expect as the concentration of your lysozyme goes up, then the turbidity, actually the breakdown of your gram-positive or even gram-negative bacteria would increase more, and thereby, because it's increasing in breakdown of lysis, you would essentially have a reduction in OD. That's what you would generally see or expect. Yes, it is an indirect method of accounting for bacterial growth, but the SPECT20 would be hopefully able to take note of the change in absorbance from live cells versus essentially past cells. So those cells that lyse accordingly would or essentially disintegrate, they go down into the bottom of your cuvette, and so likely you would expect to see a decrease in OD, and that's actually what you see from the Michaelis-Menten curve shown here. And so the beauty of this is that when you would have done this, perhaps if you want to take this course or if you want to redo this course in the summer, you can for the last exercise maybe. But the idea is that you would have done this and essentially across the different conditions, and bear in mind the units, right? For the concentrations, you would essentially plot the values for OD, and then inshallah label accordingly what you would expect to see following 1 minute and 3-minute incubation. So think about it like this. You would have done these readings at 1 minute and then 3 minutes. You could have kept them at the spec 20, but the idea is that you would record those values and then thereafter plot them one for at 1 minute, the lysozyme assay, the Michaelis-Menten curve, and then another at 3 minutes. And so you would be able to hopefully compare those results. And does anyone know what they could kind of expect to see comparing the incubations of lysozyme at 1 minute versus at 3 minutes? Both would decrease, but which more? So what would you expect to see? Yes. Like in what way? Turbidity? Like, so let's say this is 1 minute. Would it be lower at 3 minutes or higher at 3 minutes? Lower, right? Right. So lower in comparison to the 1 minute curve. So you want to be specific in that regard because yes, both decrease, but which one decreases more than the other, or providing a comparison would be very important for your other assessments, even other practicals. So, and then don't forget a detailed figure caption. So I have mentioned this for your post lab from like earlier this week, I think on Sunday. And what I do want to say is that if you can, before you end off today, we can even spend about, let me give you 10 minutes to do this. Let me just see if I have time, 10 minutes. So 10 minutes, 20 minutes. And then, yeah, I think we do. Okay, so let's do this. Actually, very simple. Draw out this curve, right? Has everyone drawn out this curve in their lab notebook? Following what I said in Quercus? I see some thumbs ups.
We didn't have.
But that's interesting. Some of you have actually drawn it out. So, okay, so let's take time then. Yeah. No, before they even came. So I was just curious. Okay. Let's see. A number of people did, but okay, it's okay. It's okay. What we'll do, we'll do this post lab for next week for those that are going to attend the review, and we'll talk about that afterwards. Or you could send it to me via email. How about that? Send it to me via email. I hope it is posted by now, though. If not, then we'll incorporate this with the reflective practice at the end. Let's go to the biofilm then. Actually, we'll do this now. Lysozyme, reflective. Let's do the reflective. Because there's 3 main things left. Reflective practice. Well, I have other things I want to talk about. But we have reflective practice, we have the lysozyme, and we have the biofilms. Lysozyme, I'm guessing about 10 minutes, biofilm about 20, reflective practice about 30. And let's see. Okay, so I just want to make sure that we prioritize our time. That's why. So let's do the reflective practice. I want you guys to show this to me, actually, everyone, at the end of today's lab. But I'll give you the time to work on this now. So think about, completing these different questions. Has everyone been able to see? That's interesting. I'm amazed. But let's go over this. I'll let you guys actually. I'll make the screen bigger. I'll let you guys work on this now because the other components are easier to work on at the end. So we have 6 main questions. I'm actually gonna check. I have a feeling it is posted, you guys.
Yeah, what's the most of it? That's it. Still up now, actually.
Let's see. This is good to know. So we will wait. In the meantime, like we could still continue with the exercise as you can see it here. Okay. I'll read out the questions. So in maximum 2 to 3 sentences, you can go 3 to 4 if needed. Describe the correlation between the Snyder test and biofilms. If you look back at week 9, previous week, you would have seen that the Snyder test, I think, was exercise 2 or 6. 2. The Snyder test focuses on the essential growth or change in turbidity following lactobacilli bacilli incubation. So you would have had a, I'm going to draw it out and you guys can make note. You'll notice some of these concepts are emphasized on. So you want to be mindful of the different concepts and discussion points that I've talked about, but also the questions that are mentioned in the lab manual or your prep of the lab exam. So you have the Snyder test, right? Probably recall was it an equation, or if you look back at the slides or even the manual, you would have had pH, I think it was like 4 to 6, let's say 4.3 4. And then you would have had dextrose slash sucrose, or sorry, slash glucose. This was one, and then there was monopresyl green or methyl green. Nonetheless, it was a dye that was green. Let's say green. Green. And so the idea is it would have started out more forest green. So it would have looked more green here, right? But the more lactobacilli that you have, because essentially the greater the bacterial fermentation of the sugar, which is dextrose or glucose, that greater metabolism or greater fermentation of the sugar would lead to a reduction in pH. And that reduction in pH, which would be optimal for these lactobacilli, would essentially also influence the coloration of your dye. Because some dyes, like phenol green, yes, but from the MSA plate, there's phenol red, I believe, or the McConkey Agar plate. Either one has a red dye. And so the idea is when there's a change in pH, then you're actually differentiating the color, essentially the color changes from a dark green hue to one that is more yellow or yellow-green. And then the idea is that, like for the conditions that you have here, there's other ones included in the manual, but for a pH that's generally low, is that I want you guys to think, is that a selective or a differential component? Selective means that essentially you are almost like selecting artificially bacteria to grow or not to grow. Differential refers to essentially having a gradient or spectrum in which bacteria are able to grow, survive, and are able to be readily characterized, i.e. I see a bacteria here and it has red colonies, or it looks like it's showing beta hemolysis under the blood agar because it's fully yellow. Whereas on the other side, I see gamma or alpha hemolysis because it's very tainted yellow and is really indiscriminatory or indifferentiable. So you have like the ability to differentiate, that's the differential components, or that's supported by differential components. And then when you have specific conditions that certain bacteria could either grow or not grow, or conditions that are harsh or extreme, those are normally selective. So I want you guys to think, is this pH selective or differential? Selective or differential? Actually selective. Because it's, think about, remember your environmental conditions from week 8 or 9? Week 8. So when you guys started off, like looking at the different temperatures, pH and salinity, pH, this is an acidic environment, right? So normally only acidophiles will actually grow here, meaning that let's say you had hundreds of different types of bacteria all in the single tube with a pH at 4.4. Then you know that only the ones that you grow or that you visibly see show a change in color would be responsible by the acidophiles. In this case, we are assuming they are lactophiles or lactobacilli, which, by the way, morphology, broad shape, right? And they actually ferment pretty much sugars like lactose, dextrose, glucose, etc. And so next one is dextrose. Speaking of sugars, is that selective or differential? Think of what it would relate with or correlate to. Yes. Yes, it is correct, and it is actually differential. So think about the consequences or the presence of your student component in relation to others. Like for example, you have maltose, I think is for mannitol for the MSA plate, Mannitol Salt Agar plate. Mannitol with phenol red would couple together to give you differential, like a differential spectrum of having bacteria that still grow, but ones that either ferment or don't ferment mannitol, right? So mannitol as a sugar, you want to keep those details in mind. I think actually, mannitol is also a salt. You have NaCl, which, by the way, is another condition, and that's selective, right? Because as we've touched upon thus far, the salt, by the way, of NaCl 7.5%. So think about the environmental conditions. Think about what I've touched upon, right? For pH, you have acidophiles, mesophiles, or homeophiles, and then the alkalophiles or basophiles. Those are for pH. You're at either spectrums or in the middle. You have temperatures for hyperthermophiles, pyrophiles, or sacrophiles, and then your mesophiles. In the middle, sacrophiles are at the colder regions, and then the thermophiles are at the higher regions. And then the salinity refers to osmotolerant or non-osmotolerant, as you see from the manual. But you have halophiles, which are generally more osmotolerant, and you have the non-halophiles, which are generally less tolerant and more sensitive to changes in osmotic pressure / osmolarity. So keep that in mind. The dextrose and the green, like the dye, as a hint, those two, like your sugar or your nutrient coupled with your, as long as by the way, the nutrient shouldn't be something that is like, let's say for an auxotroph, essentially a limiting factor for their own survival. So these like nutrients you're adding on, you're assuming that within your plate, for example, you still have LB, right? Because you need LB for bacteria to grow, right? Like from what we've done from the transformation assay with LB, ampicillin, arabinose. Ampicillin is a selective marker, right? And then arabinose is a differential one. Technically, yes, technically a differential. It is a bit confusing in that regard because technically it's a completely different setup with confirming the expression of a fully functional plasmid, PGLO plasmid, that would lead to fluorescence. But nonetheless, these two are differential. And then this is selective. So just bear that in mind. Now, when it comes to the question, right, you want to compare, and I want you to think about this when you do the biofilm assay today, because there may be some connections that you may find as you, once again, notice the differential nature of both tests. The second question is about Snyder's deep agar. As a selective medium, differential, or both. So that should actually help you out, right? So what I've just touched upon. Question 3. There's 3 different differential tester media, which you actually have right in front of you, and indicate how they distinguish between organisms and name a bacteria, genus, or species as positive for each one. So you guys could do that. Believe it or not, these questions are quite like easy for you to understand, but I actually want you to connect the dots, right? Think about the other tests that we've done in the past. And then when it comes to the 4th question in real life, right? This is where your FLR also connects. I want you guys to talk about extensions, you know, just as how you would talk in elaborating your limitations, how they could be resolved. Describe in depth a bit about a real life application for UV exposure. It could be about sunscreen. It could be about, you know, X-rays. It could be about the effects of essentially UV irradiation on cancer treatment, you know, and the growth of certain bacteria. These are just examples, but you guys can be able to, you know, come up with other ideas as well. And then we have the lysozyme enzyme itself, why it would be more effective on gram-positive bacteria, which we just talked about. And then actually, the last one I want you guys to really think about. I want you to think about it a bit in depth because there is some important relations that you guys can make inside with the skills that you've gained inside the lab to the skills that you could take outside of this lab. So think about those details. I'm gonna, if you guys haven't maybe answered the questions, let me know. But in the meantime, I could save ourselves time by going back to the lysozyme assay and having you guys, lysozyme biofilm actually, we'll go to the biofilm setup for today. So I'll leave it here. Let's go to the biofilm. If you guys are ready, I'll go over it now. So in each group, you have, don't forget these second techniques, by the way. Put down your benches. Each of these should have, and you guys can also take photos if you want on the slide, before it gets published or once it gets published. You guys already suspended? Okay, so now I want you guys to 3 points. So I want you guys to look at your tubes. You have positive, so the lab tech actually brought this one of the subs for today. And essentially, you have D subtilis with labels at P30 for 3 to 30 minutes back incubation. And sorry, you don't have to use like that. I think it's just the label. But the biofilm nonetheless has your bacteria, right? You want to suspend your bacteria. And then the other tube is of your media, the media that was used to grow the bacteria. And do you think this would be a positive or negative control for biofilm practice? This one. Positive or negative? Yeah, positive. Positive or negative?
It's the negative.
Negative. What's negative? Remember, so this is for biofilm, right? So do you guys, has everyone looked at this slide? Like everyone knows that needs to be, yeah? So I'll just walk you through the biofilm prep. It's relatively simple. You can also see it in your manual, but the idea is that this is your negative control because once you do the washes and use crystal violet of your media, you shouldn't really see anything, right? There's no bacteria. Whereas here is where you're going to have your bacteria, which should already have maybe coated the surface of the tube, right? So hopefully, once it's time for you guys to get ready, inshallah, I'll go back to that slide for the biofilm for math 3. You should be able to essentially follow very simple steps. You are going to place the tube to the right, right, the fluids themselves, and then rinse with PBS, I believe, twice, and then that could be done for about 3 minutes. 3 minutes should suffice. Pour it down. and then you're going to add crystal violet for about 10 minutes, and then let the result settle following drying. You're gonna rinse it off, the remaining crystal violet, but then you're gonna let it air dry, like invert it for about 5-ish minutes. I'll walk through the steps again in due time, but I just wanted to let you guys know this now. So that way it hopefully also helps you describe your answer to question 1. Because we're using crystal violet, right? And I want you guys to think about the relationships between this stain and what you've done from the gram stain from before to what I've mentioned here about selective versus differential components, or even other mechanisms that I've described, and other connections that you can make between the two tests. So think about the different connections that you've been able to make. Let me just see if I can open up slides. Yeah, so these were the points for the slides that I really wanted you guys to make note of. And that's why I was saying, the different components and different characteristics. Please bear these details in mind for your lab exam, or like even just your understanding of the material, or particularly. And so this was an example of the Snyder test that I was going over, because the differences in concentration of the lactobacilli can influence the differential, or like the change in color that you see, kind of like titration, if you recall, for like organic chem or chem intro chem classes. So you have a change in color from like dark green to turquoise green all the way down to yellow. This could even actually be contaminated, in fact, because of the change. The color is normally actually forest green, kind of like here. I don't know if it's hard to tell. But nonetheless, bromocrystal green. Actually, it does have a bluish tint. So this could be like your control all the way down to the darker presence of like lactobacilli. Any questions? No questions? Yes, one. So it is that's correct. It's a mixture of both. But you want to keep in mind that essentially you're starting off with a selective tube, or like the agar. Actually, I don't know if it is liquid, yeah. So it's the idea is that you're starting off with selecting only bacteria. Like the bacteria that would grow here would only be acidophiles, essentially bacteria that grow at acidic environments or at low pHs. And so the idea is that even though this is at 4.8, right, it's relatively low. And then the idea is that following fermentation, the greater the amount of lactobacilli in your teeth or around your salivary glands, the greater the number of lactobacilli, the greater the lower the pH. And the lower the pH reflects a change in color, in fact. That's quantitative, so you can see, like it's as shown in the previous, on this slide, changes in color are differential. So the hint is that your pH and, like, generally speaking, specifically extreme conditions, harsh conditions that would make it like difficult for bacteria to grow, would generally speaking be selective. So think about what I've talked about before: high pHs, low pHs, high concentrations of NaCl, or high temps, low temps as well. These are good examples of selective components of an agar, of a plate, of a test tube, even of a stain, right? So as we did from, the capsule stain, if I'm not mistaken, or sorry, the endospore stain, you use hot water, right? So really, I mean, technically your bacteria would have already been flamed. But if, let's say, actually the stains more or less focus on differential components, which is where the other, the dyes come in, homocressyl green, with the supplement of dextrose or glucose included. So once again, these two are differential components, and this is selective. Any other questions? No questions. Let me go back. So can I just get a follow-up? Has everyone done the reflective practice? Yes, but from the time that it was on, did anyone, you guys are still waiting? Okay. How about this? Then spend the next 8-ish minutes completing the rest of the reflective practice. It could be brief. And then we could describe question 6 after. So spend about like a minute or two per question. Based on what we've discussed, pretty much most of the questions are already answered.
I was trying to show you the finger caption. The sweet Agar of videos over liquid culture fluids and ice cream. Yeah, is a overnight in liquid culture. Oh, okay. Okay, thank you.
Well done. If anyone else has to just confirm their lab notebook with me, they can. If there's any questions.
Oh, you wanted a week 7?
What did I speak with you earlier? I don't
Oh,
recall.
no, I came up and checked that and you were like week 7.
Yes. I guess it's supposed to. Or
Wait. Sorry, this is week 7. No, this is week 7.
should live or not. I think this is what I was looking for. Can you describe this is actually, I think, is technically up on week 7. But it's really week 6. It's not. Could you describe a bit more? Okay, that's what I this. I wrote it, but then I had a discussion for this. That's okay. So this. That's good. Okay, so just write just like that. Like that. Let me see. Did I confirm your week 6? I don't know if I wrote it down. Yeah, or week 6? Oh yeah. Okay. Just okay. So you should be okay. 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 This. Okay. Yeah. I did check. Yeah, I checked. Can you see this one? Yes, okay. And then you can go backwards.
And then continue here.
You should be good. So far. Yeah, good. So, Yes.
Just mixing the process when you do that process with the flow. So this is the negative control, positive control. This is
Do you know the unknown that you have?
I think I can do. This is the this one. So this helps.
Yeah, perfect. Okay. Perfect. I appreciate it. Nothing else. That should be good. Great job. That would be weekly.
Yes, so actually, this is our place that we have to manipulate and do it.
Could I see week 7 intro as well? Week 7. Maybe a bit more. So,
It's observation.
I didn't mention this, but you also need to do. So,
So these are the not the caption considered.
the captions we do. I'll help write an example. So this is figure. Think about your method. Figure 1.
Yeah, I mean.
This is for this is for
Viable count. It's a viable count for generation on assignment we did it.
So determinant determinant I'm just gonna write this much, and then you can be able to continue the rest. So generation time of E. coli at 37 degrees, and specifying the details of how you were able to do this using the viable counts or the viable count method.
Okay, then like this much I have to.
But that level, the detail, you wanna, okay, sorry, continue actually.
I got your point. So with those, all the figures I need to make the caption in terms of what we are doing.
Yes.
Not just the label, what it is.
Yeah. You want to have like specific details, having a title, talking about what the overall message is of the figure, then your methods briefly or the conditions that you have your that you're accounting for in your result, and then the brief summary of the results. Okay. Actually, let me just make sure I didn't say the wrong way. Post lab weeks. One moment. One moment. Oh, perfect. I appreciate it. Great job. Okay.
That's how it's going on for the process of the discussion question answers. Observation we had it in the lab and the procedure which we performed in the lab. Discussion and conclusion.
Very good. Apart from the last part, just make sure that your figures have a look back at any other ones that are like plates that need this space.
Then you can make it.
I appreciate it. Thank you. Yes.
Oh, I just asked the class data for
That's good.
yeah.
Perfect. Great job. Great job. Perfect. Okay. I'll get both of you behind and yeah. Okay, check. Yeah. Make sure I check you. Nothing. That shouldn't be it.
I should do. Do we use the beat? Right? I'm not missing it. No. Okay.
As long as you just keep in mind, though, I told people this before. I'll say it out loud. Folks, please remember that your lab notebooks you want to bear in mind that your figures are very detailed, right? Like for your FLRs, I actually am taking note of this. So that's where you just have to be mindful that you have specific details in place. Is this for the reflective practice? Perfect. So I would recommend, not now, but at the end of today's lab, we'll go over the reflective practice just so that way I could check you guys up with the post lab of as well the plates. So just have the plates, figure captions, the gelatinase test, and then those three components should generally suffice. TDT, you guys can show me whenever you can. The sooner the better. But I would say focus on what I've discussed thus far, and then that should suffice. Anything else? Yes. So like think of it like this. Okay, one more. If you let's say this is your FLR. Okay, so I'll just describe it. Intro, so your FLR will have like an intro spin-off or like a primer on what your aim was and how you undertook that aim. So that's or your objective. And so pretty much describing that as your first component, and then within that, refer to your first figure. Figure one, which would be your full plate, or your figure of all, I guess, 28 plates, if I'm not mistaken. From essentially all four conditions, like all four bacteria, and from the four bacteria, all time points for UV exposure. And that itself is gonna take a bit of time for captions. So please bear that in mind. Figure captions are pivotal. And then thereafter, you describe, even though it's a summary in your caption, in your results description thereafter, you're providing more information. You're describing what you've seen across time points, what you see comparing across the different bacteria. So note those differences in mind, and then refer thereafter to your table. And that's where you'd hopefully describe the changes that you see on a scale from 0 to 3, which you need to mention in your captions, the change relatively in bacterial growth across exposure time. So bear that in mind. Bear in mind units, right? We're in minutes. And bear in mind that you're placing close feed into the details I mentioned from Quercus, and also when as I interact with you in other modes, like as we are today. So just bear that in mind. If there's questions though, and maybe things that we could rectify early on, it'll be better now than later. So just bear that in mind. Yeah. Did that answer your question? No, no, they have to be, so like, I actually do take off marks if it's not like formatted properly. So bear in mind that you have results. The results should be immersed with your figures. Kind of like you shouldn't leave them at the very end, because hypothetically, I would have to go up and down to see the relationships between your table, your figure. And it's not even just about that. It's about you delivering a unique story about UV exposure and the effects on bacterial growth across different bacteria. So you want to focus and like deliver in a nice formatted way how much effort you put into the FLR. So I would recommend having it in that way. No problem. Is your issues also in there? But I will come to you guys. At the end. So the same question.
2 hours ago, because I found something here. I just wanted to add one for this one this lab intro. And I just wanted to ask if there was anything else that we were missing for our lab. You didn't call it earlier, so we just wanted to make sure.
If I haven't, then no.
Okay.
I'm just gonna look, but I don't believe so. Julia, you're good. And Marissa, you're also good.
Originally this time, because after my email with you, I figured out my email this time. But thank you for that as well. I was wondering, the table is just gonna be the numbers, or should we also put a little bit of analysis on each, on each one? Should we like describe the growth, or should it just be numbers with the with the table caption, describe what they mean, and then in depth in a paragraph afterwards?
Pretty much in depth in a paragraph.
So just the numbers.
The numbers in the table, if you could provide data analysis, that would be amazing. But I don't want to rush you if you have other things.
No, I put the table and it looks really ugly. And
Don't
it's
worry.
really bulky.
You can show it to me, and I could.
Yeah, I'll show you
That's
after.
okay. That's okay.
Thank you.
No problem. So folks, yes, like I take into account your effort, right? So if you, for example, put in a lot of like effort and put a lot of detail into your like captions, for example, your labels. So for that context, I sometimes like take into account that maybe, you know, like those different pluses are things that you want to essentially make full use of, so it shows that you've put in the effort that is needed into the course and into the FLR itself. Are we done now for the 6 questions? Please raise your hand if you're not done. If you're not done the reflective practice, please raise your hand. I want to see how many people. Okay. Let's wait 5 more minutes. Is that okay? Yeah? Okay. Yes.
If you keep talking about the detailed manufacture, I want to make sure so I was going to include their as it was, and I was going to use a table language. I was going to say and I was wondering, does didn't talk about the growth in the caption, we talk about the growth in the actual results, and we reference the big shot and like there's 5 more growth.
So just to clarify this for the figure caption again, you guys are gonna have a title for your observation, and then for following that, you're gonna talk about the conditions in which your plates have been set up, i.e., the different bacteria, right? Mentioning all 4 of them, mentioning the changes in time, the different increments, and the units and minutes. And then after that, and actually bear in mind that you have 2 sides, right? 2 sides of the story, 2 sides of the plates, A and B, and what those 2 represent. Following that, so that's like your methods component or portion of the caption. Thereafter, you're having a brief summary of those results. So showing what you've seen across trends, be it from the different bacteria or across time. And so you want to think about those, and then those 2 can be 2 sentences, but then following your figure, you're going to describe both of them more in depth, i.e. changes across time and changes across bacteria in terms of the effects of UV exposure. So bear those details. You're looking at this in manifold ways, so you want to be as descriptive as possible. And I won't take, if you're slightly off the word count or a page limit, like half a page, I don't mind. But bear in mind as well, your results, like the figure captions and the figures themselves, are not included, right, in the word limit or page limit. So focus on, delivering the details that are required, and hopefully that should support your endeavors. Any other questions? Yeah, did I answer your question? Perfect. For the practice? Oh, for the figure caption.
Are you checking the
For
Mannitol?
yes. Figures. Everyone's covered.
And then I just looked at this for this. I was a bit confused because when I do it in the class, like it's like this, right? Because it's a figure of a curve. So if you draw a line that's straight, it would be off. You know what I mean? Like, as I said, this is exponential, so it should be linear, but then these points are exponential, so they're like flat, and some are log of flat.
It's a bit interesting because the reason I bring this up is that technically we'd actually have it essentially balanced. Essentially all supported by a single.
It wouldn't, because generation time should be during the exponential, right? But like, if you do that, it would be skewed. Because like I was confused because in the instructions for the absorbance they didn't mention line of S-fit. That's why I didn't. That's what I thought too. So like, because like you could only measure next exponential.
You know,
It would be skewed. Or like,
I will take this into account because you have actually labeled them.
Yeah.
But I do want to question you because technically speaking, just for example, you actually want to have
so like
entire
here to here.
generally.
And the same for here too. Like just like
yeah. Because okay, let's have a look at your generation times.
Because I know from B12 they were saying like it should be during the exponential.
That is correct.
So
but it was assumed that it would still be log base. I'm just trying to understand though. It could be also the representation of the axis itself. Because I've seen others that are like continuously linear. So let's have a look. It could be maybe the stepwise increments themselves. But I'll account for them. It's pretty good. Okay, so that way I can adjust. What's your goal? What's your
To think of my group members that you want
let me explain some. I don't wanna like vocalize, but technically speaking, one should be from others.
and we didn't know.
That's okay, that's okay. So my advice is continue to answer more questions. Try to, especially even though we're almost done, you still have next week that you can engage more. And I would recommend, and I'm basing it also on like the proficiency during the lab. So being able to like essentially, it's almost like semi-evaluations where I'm eyeing what people are doing and then confirming like with them as they communicate with me what they are doing and what their next steps are, what their questions are. And generally as well, like, this is a spectrum. So people could be at the lower range, you could be at the higher range. And I did mention this as well, but C2 and D1 overlap at 98%. So technically, you don't know. But I would say at the same time, don't lose hope. I would say continue to do more and engage with me at the bio. Does that make sense? And ask more questions. And like seeing. Keep it up. Keep it up. You're doing great, by the way. Please know that. Great job. Folks, I just want to confirm, are we all done the reflective practice? Okay, if it's okay, I'll lift my yours briefly. But now we're going to be starting the biofilm. So I want you to spray down your benches. I want you to essentially start getting ready the biofilm prep. And then inshallah, you could start on the gas for the Bunsen burners. So start off the Bunsen burners, the gas is on, and you guys should be able to get ready for the biofilm prep. Perfect. I'll just make note of it. I don't know if not anything. Yeah, this is week 6. No. This is week 6. I think it was yes. Yes. Later would be better, actually. Later would be better. Is that okay? I really appreciate it.
Do you need this anymore? No? Okay.
Thank you so much. Okay. So let's go back. The reflective practice. We'll do it afterwards. We could see the reflective practices afterwards, folks, but I want you to focus on today's lab. Try to finish it in the next, I would say, 18 minutes. Very simple, but there is a time point you have to wait for about 10 minutes. So I want you to just bear in mind the following. There's a bit of waiting, but what you're going to do is essentially turn on your Bunsen burners, right? We're still doing aseptic technique all the way to the end, and that's something you want to keep in mind. Once your Bunsen burners are on, you should hopefully have your two tubes. One that's your negative control, one is of your bacteria. And so the goal is you're going to dump the two of them into the Wescadine, right? Both of them should have their bacteria present. Let's see if there's any other things. Mix slant into, oh, I forgot to mention, you guys should resuspend the tubes. Did you guys resuspend them? Yeah? Let's actually pause. I don't want you to dump them as of yet, because this is an important point. Because you don't want them to just settle. Keep resuspending. It may take a bit of time for the biofilm to the pellet underneath to essentially resuspend. So I want you to do that just for the non-control, the experimental condition. And I'm just gonna briefly show this as well. Be our final time. You look at week 10. Oh, sorry, week 9 material. Mannitol is a salt. It's both selective and differential. Yes, there is. So what did I miss? Oh, sorry, biofilms. We're gonna go up or down technically. So you would have a fresh slant of Staphylococcus epidermidis. That's what you have in your biofilm, and we're skipping to lab 2 essentially. So I want you to, resuspend and then decant or essentially place both liquids, your control and your Staphylococcus epidermidis, into the Wescadine. Make sure, by the way, you flame your tubes before doing this, as you want to be mindful. What limitations could it give? Yes, Raj.
I'm gonna walk. Yes, but I think like one big
I want everyone to take out their so wait for to get some. So let me see if I can get other pipettes for you guys. For the time being, dump your tubes into the Wescadine after you suspend them well. You should see the pellet disintegrate as it lifts up and resuspends into the liquid. You just bear that in mind. Try not to move it up and down because technically your biofilms should be intact. Just resuspend by yes. Cool. I want you guys, it's getting a bit of a funny task, but you guys are gonna actually, I want you to have some pipettes, but you guys would actually manually place them here. So maybe, one group at a time. I'm gonna see if I can find plastic pipettes, but in the meantime, as every group decanted, their liquid combination. So decanting means that you're taking them out and putting it into the left side. It's all both of them in the left side. You guys should be mentally careful with this about it. So in the current time being, you guys can move forward with the PBS. Try to let groups go with efficiency. Actually, I'm gonna here. Let's do one concept. I'll use someone's Bunsen burner. Part this read online. This is very crude, meaning that we're doing very simple observations. It doesn't need to be bigger. It's okay. I'll hope that
Oh, I don't know. I didn't. It's like, which one's the control? Yeah. This is control.
the next groups to come in, single file.
Go stand in a line. It's right here with the light. Like,
If your group is ready to use the crystal violet, they can take it, but I would just wait a bit. For your to draw it.
Just like the other patterns in YouTube, while you.
Everybody's ready for Anisha, I believe you're ready, right? You can get ready for the first violet stage. And then I want you guys to look back and record your observations for the gelatinase test, which is another one. And I think there are two groups that are missing there. So try to take them. Who's ready or done with PBS washes? They could come by as soon as they're air dry, which could be about like 1 to 2 minutes, or even 3 minutes. Let's open air dry, otherwise the moisture could be retained. You guys have they don't want to drag it. So if your group is done, air drying their tubes inverted, lids open, for about 2 to 3 minutes, they should get ready to actually start adding in the crystal violet. Let it settle for 10 minutes in each of your tubes. And then once you're done, we'll rinse them off or even air dry directly. Rinse with water and then air dry. And then you would actually, as it air dries, see the results quite readily. So then thereafter, I want you guys to just record those. You could just write them down in your lab notebook. It's very simple, right? Because like you could just write it. You don't have to make too much, like describe too much in detail, but just observe and make note of what you've seen in your lab notebook. That will likely be the final. If you've done, by the way, the gelatinase test results, if you've recorded your results for the lab notebook of your plates, of the reflective practice, of the gelatinase test, the TDT results, if you have the time before next week, that would be amazing. If you have all of that done, you can show that to me in the next 4-ish minutes. I want to just make sure the crystal violet's good for you guys. Oh yes, and let me go back to the slide. So once we're done, like the lab, then you guys can like spend this time, enjoy this time, you know, because yeah, you likely are only doing BIOC17 once, right? So, or at least like consider, you know, the beauty of these different organisms around you. I'm a cell biologist, so microbiology is still, you know, applicable indeed. Yes. Oh, I was saying just wait for 4 minutes. What I want you guys to show me. So let me just say it once again. Actually, I didn't say this, so I'll go back. We've talked about the lysozyme assay, right? I want this to be done before you guys leave, right? So here's what you need to do. I'll write it down, or I'll say it and then you guys make note of it. The plates, right? The plates, raise your hand if you've done all 3 plates in your lab notebook. Are you guys recording the results of all 3 plates with figure captions? Figure captions. So you need to be done that. That's number 1. Number 2 is the results from the gelatinase test. Have that also written down. The next one is a reflective practice. I could see some people playing around. I don't mind. I'm also a student. So okay, you have 3 plates, gelatinase test, the lysozyme, sorry, the reflective practice. You have the lysozyme, which we're going to talk about now, and then the biofilm is going to come up. There was one more, thermal death time. So the thermal death time can be done because it's a bit more elaborate. It could be done today. It could be done or sent to me via email next week. But I want you guys to show me as much as you can. And speaking of next week, actually, this is where I want to talk about your lab prep. I'm doing this out of my time, and I do want you guys to attend for there being benefit in going through the past 10 weeks. And I'm splitting them up to be like weeks 1 to 5, half an hour per week, inshallah, per review. And that will be, I'll write this down. Hopefully it works. Oh, it's coming out Tuesday. Okay, Tuesday 10 to 1. And then Thursday from 10 to 1. Sounds like the baby's cute. So 10 a.m. both days, so Tuesday and Thursday, 10 a.m. to 1 p.m. How many of you have exams during that time? During that time, please, by the way. I need to like take that into account. During that time, okay. And then your exams are like 2 to 4? 5. Okay, so let me see. I'll send an updated time in case, like, I want to make sure that you guys have the time to also study for C17 and also to, like, I want to make sure that you guys have full concentration and full awareness of what's expected for the exam. So nonetheless, you do have a review with Dr. Brent on April 4th, so this Friday. So try to attend if you can. Yes. Yes, they will. But when it comes to participation and upping your grade from like a C1 to a C2 or C2 to a D1 or D1 to a D2, then that's where you actually have to show me that you guys really mean what it takes to essentially up your grade, right? Because I'm trying to be aware and I'm trying to help you guys out, but you have to also put in the effort And I'm aware if you guys do. So just keep those details in mind. Also, please like understand that I have a lot of like emails. So as you can see, I get notifications often. So please just bear that in mind. If it takes a bit of time for me to respond, just be aware of that. Also be mindful that I will only be accepting emails up to like as ideally up to today. I don't want you guys to like send me too late emails because at the same time I have other practicals like in the upcoming days. So just bear that in mind. Any other questions, final questions? Yes.
Average for each time point.
Just one time point. And to make this easier, everyone write this down. For the microplate reader, you are taking the values or the trends from columns A and B for 50 degrees at the first time point. So let's go to, so yeah, the first time point, and then the last time point is only from row C for 65 degrees. And that's where you're only going to plot out the results and determine the curves for 55 and 65 degrees up to 18 minutes. Column A, by the way, is the blank. So you would technically need to subtract the values accordingly. So bear that in mind as well to correct your absorbance readings. And then thereafter, actually, yes. I'll repeat it. So you have for the TDT, like from the microplate reader, you have different time points, and to be reliable with these results or aware and cognizant of the how they appear, I want you guys to take the 50 degrees reps. There's two of them, rows A and B, from the first time point. That will be one curve for absorbance readings across time. The absorbance readings need to be corrected by the blank, and that's in row A, or column A, for both across time, for both temperatures. And then, so you're plotting the absorbance readings from there, and then that are corrected once again. And then at 65 degrees, you're doing the last time point, which I believe is at 4 hours. So this goes up longer, but you should be at 4 hours post-incubation, be able to take measurements of 65 degrees from row C across time for the OD readings. Does that help? Yeah. The last time point is around 4 hours, 4 to 5. It's just the last time point you have to worry about, nothing else. Yes. No, so the microplate reader is not 0 to 5 minutes. The microplate reader is every 3 to 4 minutes, essentially. Maybe I'll show you the slide. Yeah.
So you mean we're plotting every 3 to 5 minutes?
Every 3 to 5 minutes, but so there's different tables. The microplate reader takes, like it's actively incubating across time points and recording those observations or absorbances. The idea is that from the first table of the spreadsheet, you're gonna actually take recordings at 50 degrees, right? Because at 50 degrees, it's gonna go for each column, each cell is gonna have a different time. It's gonna be 0 minutes, 3 minutes, 6 minutes. It's gonna go up essentially across time from the time that I was here. So the time that we were waiting, 50 degrees, 65 degrees, it was heating up. I would place it into another well, You'll see this in lab 8 live. So if you want to have a look back, you see the outlines, but it's pretty much also there. And you should be able to essentially record the observations of absorbances in that way. I could also return back to it if you need help.
Yeah.
I could. That's okay. Yes. Yes. Column A. Column A. I could also go over this. I could go over this in an email as well. Just give me a bit of time because I'm also like marking and stuff, or like doing other things as well. So it's a bit, it takes a bit of time, but nonetheless, complete everything else. And let me know if you have any questions. Yes, I will do it. When you guys are ready, click the button, you can go straight ahead. And then I will just briefly share. You can bring up your reviews for the button. In the meantime, you can turn up prevention. Well, I think I'll say close them up right away. Just a while now. That's okay, but has every at least brought to meet their recruits for this pilot patient? I think that's one group. So next one, are you guys okay? You have someone. You have crystal violet, right? Is you inverted or you're air drying right now? Okay. Perfect. So yes, I'll explain that now. Is that what you're also probably?
You said one time. Or then you said the beginning of the end, just the first and the
Just the
last.
first and last table.
Okay.
Let me just highlight this. Okay. Okay, so can I have a show of hands of how many of you are waiting for the biofilm to be completed? Waiting, okay. So in this time, let me just let me answer Ms. Cora's question. What's your question? Yes, I would like to have. Is that okay? Okay. To make things easier, you won't have to show me your lab notebook, but I want to stress that you need to have the post lab done of your 3 plates, the gelatinase test, and the TDT. I want you to send to me via email. I'll talk about that in the next 4 minutes. So I'll look at this after. I'll look at it after. Perfect. You can leave it here. I just want you to focus, folks. Let's pay attention. We're almost done. I want you to bear in mind that you have the results that I mentioned, the TDT, the gelatinase test, the 3 plates, the lysozyme assay, which essentially for timing we can't do now, but I want you to be able to essentially take note of the different saliva samples. These are essentially, if I'm not mistaken, They were supposed to be at specific values. Yes, this is at T1, like at 1 minute, and this is at 3 minutes. I just don't know if they don't show any specifics. So I'll confirm with Dr. Brunt. Yeah. So pretty much what you would have done, and this is something I want to bear in mind, is that you essentially would get these values, right? These are diluted, and like from 10 to the -1 to 10 to the -2. You could take photos, folks, you could take photos in case it's not sent in time to you. So the idea is that you're going to take a photo of this. I'll try to copy this and place it here. And then you would interpolate. This is a bit hard to do, right? Because, I mean, practically you don't have the results with you right now. But what you would do is essentially reading off these OD readings, you would interpolate. Let's say we're looking at 0.43. Hypothetically, 0.43 would be somewhere around here. And then it would give you a concentration of, let's say, 800 nanograms per mil. Now, remember, this is the 10 to the -1 dilution. Meaning, how are you gonna get the original stock solution, or essentially original concentration of your saliva, or the lysozyme in your saliva? What would you do? Divide by, okay, divide by. Yes, so divide by the dilution factor. So you're dividing by 10 to the -1, which is actually multiplying by 10. So if you had 800 nanograms per mil, what would it be, the stock or the final concentration of your lysozyme in saliva? If it's 800 nanograms, what is the final concentration? I want to hear from everyone. 8,000. Are we 100% confident with this? That's correct. So keep it up. So I'm going to ask one other question so that way we can just make sure we're on the same page. So the lysozyme, I just want you guys to understand this, try to draw it out, and send the lysozyme with the TDT results to me. Via email is okay. You don't have to show it to me today. I know it's a bit tough for timing, but let me just remind you. Yeah, I'll send an email describing that in due time. Any questions? Let's see. So you guys are waiting. What's the time like for you guys? How many more minutes? 4? Perfect. Okay. 4 minutes, 3, 4 minutes. Okay. Let's spend this time reflecting. Number 6. Oh, look, I could add some nice colors. Okay. Okay, so let's look at number 6. Okay. I want you to really think about this. Hone in. Think about for a moment. Close your eyes. Don't think that they're like, imagine, hopefully your Bunsen burners are off, so that way you can close your eyes quite easily. I want you guys to think for a moment, what have you gained from BIOC17? Yes? From me?
No.
It's okay, it's okay. If you find this as a technique, okay. You guys, you can share some more ideas, yes. Yes. How to calibrate a microscope, right?
How to fix bacteria with slide.
That's amazing, right? Coagulation of bacteria, isolating colonies, spread plating, PFUs. Yes, someone? Yes, yes, yes. Aseptic. And that's actually something I try to impress upon, but yes. Detailed figure captions. Anyone? Semi-log plots. So you see, like, there's a lot of things that you guys are learning and a lot of things that you want to be aware of for the final exam, right? It's 1 hour that you get for the lab portion, and there's short answer based questions, pretty much connecting the dots or pretty much surrounding the entirety of our lab from weeks 1 to 10. So the idea is that you guys want to be able to see those skills, see the results, the concepts, the topics, the methods, the concerns, the limitations, expectations of different exercises that we've done together. And I know it sounds like it's a lot, but that's why you guys need to prep early. You have 22 days from now, right? 3 weeks. Even if it means you spend 1 or 2 hours, let's say an hour and a half every day, studying for at least the lab component, that should give you a good amount of time. And the benefit of that is that you could very likely repeat your review 3 times. And I usually use this trick when I was in undergrad because that's how I'd get 90s in my courses. So just bear that in mind. You don't have to follow my strategy, and I understand for some of us it may take more time to study, and it may take for others more quicker time to complete certain tasks. But I want you to really spend some time, like focus, hone in, based on what we just described of the material, but also seeing how like these other, skills that you've actually gained could be used outside of the lab. And actually understanding the relevance of it. So like even when you're doing your FLRs, like talk about the extensions, talk about the relationships to your future aspirations. Let's say you want to get into med school, you want to get into grad school, you want to get in law school, you want to get you want to have your own lab. Like these are things that you want to consider, right? These are things that you want to apply and understand how you could differentiate certain bacteria if you have like a bunch of them on a desk and you have to describe, let's say from a histopathology reading, okay, this person has XYZ test results. Is this, so let's say it's a gram-positive bacteria. It's capsule stain negative. It has endospores. What type of bacteria would it be? Like these are things that you want to think about. Or as another example, looking at your plates for the beta hemolysis. Okay, what type of bacteria, what type of condition is this? Or what effects would that have? So, like on, let's say in someone's body, like in vivo, or like thinking about the effects of, outside of plates, data analysis, semi-log plots, generation time, expect, like these topics, like expect something, or expect, like a lot of things to be connected and overlaid upon one another during the final exam. And I think with that, I hope you guys can send me your results before you end today's lab. Or like at least you could show me, for example, the plates, whatever you've done thus far, and then show to me as well via email the rest of your lab material. And so once again, I want to go over those details, or at least half an hour per lab next week. And we'll try to find out another time. But just to confirm, everyone's good for Tuesday, 10 to 1, like pretty much this time. Yeah. I'm seeing some like emotional faces. Okay. Try to come because that's your opportunity to perhaps, see what you're missing out on. And if there are no other questions, I will end off. سبحانك اللهم وبحمدك نشهد أن لا إله إلا أنت نستغفرك ونتوب إليك. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. Jazakumullahu Khairan. Everyone, I hope you have an amazing week. Let me just answer a point from Krista. And I just want to highlight this. I try to be a very, don't say easygoing, but very understanding TA, right? I understand you guys have different paths in life, like, and maybe like this is a very stressful course for you. I want to be that lending hand, that helping hand to help you guys excel, right? And you probably can see this in different ways, perhaps. But I just really hope that if you have, like, if I've ever come across rude or stressed out, or invigorating or emotional in any way, just let me know. I don't wanna at any time make you guys feel down or feel like you regret having this course with me. I really hope you've enjoyed the different things that I've talked about or what we've learned from each other. And I hope you guys ace or excel in all of your aspirations, or that which is best pleasing, indeed. So, and if there's any questions, concerns, let me know. Let me see if there's anything else I need to give you guys. Anything else, folks? You guys can, if you guys want to go, you can. You can send me your details over email. Just make sure that you've actually have seen the biofilms, right? You've observed them. You can take photos as well. You can take photos of each other if you guys want to like conclude the practical with one another. That's also cool. Apart from that, I really appreciate the time, folks. Let me know if you have any questions.
So that's my bad. I just crossed out. Oh, this is the square.
Thank you so much.
Okay, I was going to say, but this is the like that you asked us to like write down and give up. And then this is the last chapter. DNase hydrolysis, the last page, the reflective factors. It's like liquid. Okay. And then discussion.
Perfect.
And conclusion. And sorry, I'm gonna finish.
That's what you well done.
The slide.
Great. Hey, don't forget that you actually accurate concentration. I just got to say this one more time. For the lysozyme assay, it's almost like a trick because you need to bear in mind the dilution factor. Don't forget that once you read the absorbance values and interpolate to find the concentration, you need to multiply by the dilution factor's inverse. So if it's 10 to the minus 1, multiply by 10. If it's 10 to the minus 2, multiply by 100. Bear those details in mind.
You multiply the absorbance.
To multiply the final recipe interpolate to find the concentration, you multiply that concentration by 10 in this case, or 100. No.
I feel like some graphs that you want us to do with what we have. And then discussion, conclusion, notes test, and I'll finish the biofilm later on, and I'll keep all of it from
Perfect.
okay.
Thank you so much. Great job. That's okay. No need actually for discussion. Well, it will be very beneficial for your prep for the lab exam. But if you can, send it to me if you want to also help boost your grade. If you're looking at yours, it's okay. If you're looking at others, I would kindly appreciate that. That's great. So much.
Is that the I forgot the name. Gelatinase, yeah. So I've put it here and the 3 plates with your caption. So there's 6. I can see like strand.
You want to show me this? When you guys are done, let me know just in case you have some on the results. DNase hydrolysis test should have a halo surrounding your positive result. So bear that in mind. It would be E. coli, but essentially you just want to see that halo. So the blueness disintegrates.
Oh, let's see.
So you see, it's actually less. It's actually less blue. It's actually whitish, right? So that's actually it degrading DNase. Maybe even on the table, because like the table is black, so you can see it hinted less. But that's Now you get to see it up close. You guys can take photos of your plates. You can have selfies with your plates. You can have a selfie with the incubator too if you want to open it up. But the incubator should be okay. Tubes can be left at the front, in the middle. Thank you so much. Yes. And the TDT. I'll talk about that in the email, but try to do as much as you can. TDT, lysozyme. If you are in a rush, just share the other things that I've talked about: the plates, the gelatinase test, and your biofilm. And the reflective practice. Yes. Did you see a lot of film in the negative control?
Really, but like this one's like heavier. This one like is still
Okay, that's good. Yeah. Perfect. Yes. Thank you so much. Please. Thank you. Thank you. This is fine. Okay. Yeah, it is. It's not the same. No, I appreciate it. I hope you enjoyed it.
Your announcement, you said you can either grab the Bible or the.
I know. But Dr. One, I think, wants you guys to also go read it. This is why I reserved it for the post lab for this week.
Yeah.
But technically for the remainder, which is the microplate reader, you can kind of do essentially a stuff.
Okay, so and then what did you say? First one is the blank, and then A and B.
It's also on the slide.
Okay.
I think the slide will make it easier actually. I have a lot of.
You know when she's gonna.
I'm gonna ask. Yes. No, not
Okay, because like
that's okay. Oh,
it also this is like it also wasn't so if I did a sample data, do I still have to do that one or is it?
for the microplate results are listed all that page. But this one is actually supposed to be approved.
I just
Well, technically, I just
it's not the curve like that.
but you want to actually follow through with the units or the increments because, like for example, this appears to be 0.7 at like 250 nanograms per mil. So I would say wait for the lysozyme if it'll likely be up. But in the meantime, finish everything else, and then once it comes in, you can complete the lysozyme assay. Depending on when you post the slides, I'm going to do this. You may do it today also, but the sooner the better. At least you could send me the other. Folks, just to confirm, everyone has taken their unknown assignment, right? I really hope everyone has. I'm gonna check the case again.
Sorry.
Yeah, sure.
So bear in mind, you have to clean your desk, right? And don't pay attention to me, guys. I'm just playing with this.
Ethanol.
Concentration, what concentration?
70%. 70%. 70%.
95% is your decolorizer for the gram stain. I hope you're not recording there.
No. It's my
She's showing
video.
me her video for
It's my
the.
C90 project.
Oh.
So, okay, sorry. Let's continue.
But what was the topic though?
Yeah.
Oh wow, so cool.
I didn't let any of my group members see it or access it or touch it.
Like,
Viewers can touch it.
Oh wow.
I'm a control freak.
I hope you was in my group. I don't have to do anything in it. You every step. Like this.
Oh my god.
yeah, I did not want actually you guys to use it.
Yeah.
Guys, like,
Don't put my voice on
but reason was the idea. Yeah. Now, when it comes to
It's okay. Really, yesterday I was working on that. I submitted it last day.
We did too.
There is a condition where, for example,
Because I stayed up all fucking night. I literally went to sleep at 5 in the morning. Like that's when I finished the show.
What do you by?
But technically, I just
midnight?
did that.
No, I know. Sunday.
Yeah.
Oh, yeah. Stayed up until 5 in the morning on Monday.
Some people were joining
It was same with me as
but it was
I
like,
stayed up.
or like, or they were doing line by line by line. But essentially, you actually want it to be.
I choose diabetes.
That's good too.
How long is your video?
This one I can tell that you actually tried to.
It means R is extending the limit. R is 11 minutes. Yes, and nobody else did it. We are not supposed to.
Video's 11 minutes.
If the number says around 5 minutes. I don't know. That's my group partner edited it and put it.
Actually, let me just put it back.
It is 11 minutes.
But the thing is,
They are.
Resubmit?
Let's do Monday.
Why?
Because it was submitted. They said we read all the basic instructions and it's all good. I was thinking that like all the samples they posted on the focus is 5 to 6 minutes. It's between like that time period. And our is this would be a good with it.
It's Emily Bell, but the rubric says, and she had made it clear that like they were doing things.
generally, yeah.
I guess mine are not.
The thing is,
11 minutes. That's a documentary.
Otherwise, like, okay, it depends. If your curve absorbance readings do show a plateau and do show a lag, then yes, you can incorporate like the plateaus, like how you had before. But if it's continuously showing a log, have a straight line. Yes, that's my concern. Technically, I would have a feeling it would go in log if it's a curve. That's all right. Yeah, sorry.
I think it's like.
No problem. No problem.
And then, okay.
So now we all have an access. Just read our CR and
Oh,
short movie there.
And then I'll finish this.
You have the.
You know how they do it in like the movie?
Yeah.
Run through. I have to go. I'm sorry. Julia's gonna hate me.
I just want to confirm, you were waiting in line, yes?
Yes, yeah. I mean, I've been standing here. Yeah.
You don't.
What was it? Do you, I'm sorry. This is reflective practice here.
Okay.
Six, and then these are the images from last week's.
So you know what there is like.
Yes, so it's the TDT and the license plan that was emailed to you. Is there like a time to do that by?
As soon
As soon as, okay, sounds good.
as.
Okay, maybe tomorrow.
That's okay.
Okay, because FLR is due tonight, so. Well, due tomorrow, yes, but gonna do it tonight.
That's good. That's good.
Tonight.
If you want to prioritize like the FLR, you can.
Yes.
But I would just say don't forget.
Yeah. I will. Okay, thank you.
No problem. Take a picture. I didn't see it. Are you saying about the microplate?
Yeah.
I don't have it here, but it's on it.
Oh, okay. Yeah, that's me.
Perfect.
Did you say you were gonna email us about that?
Yes, but it's gonna be like, you should have the details that I already mentioned. So pretty much first two columns of first time point for 50 degrees, the last, like the first table essentially. And then the third column for the last time point, 65 degrees. Which is relatively simple.
Yeah.
The three plates, you have a removal. Okay. Okay.
And then these are the
That's great.
Yeah.
Okay, perfect.
This one, do you know which one's one?
Yeah, I'm gonna ask Dr. Brent. It could be that they're just two different samples. Yeah. Trying to remember if one was a control from last year. I don't know if one was a control. I have to find out. I'll find out. CDT or? Oh yes, that's okay. That's okay.
Thank you so much.
No problem. You too. If I don't respond like in the next 12 hours, send me an email, please. Yeah, I'm okay, maybe 6 hours or 8 hours just to be safe, because like I will be swimming, so but that's a whole nother thing. But anyhow, because I'm training to be a lifeguard, so that's another thing. But I don't wanna I don't wanna deter you guys. So yeah. I was worried that you're in a rush, that's why. Okay, if you can wait like maybe 5 minutes. Yeah, perfect. Okay.
The caption you want me to do it from the sample.
No, like.
This is post level observations, post level something like that. You asked me to put the detailed caption, which I did, right? I did it for everything which I have. This is today's interpretation. I put the detailed caption. Let's see. This is also when we discuss questions.
Any questions?
Yes, I'll email it to you because I'm unable to attend. That's also why I have to complete. Thank you.
No problem.
Hi. We mind discussing like the 3 plates and everything, the 3 captions.
You have the gelatinase as well, or?
The gelatinase test is here, this discussion.
Oh, you wrote it down.
Yeah.
Okay, and there's one more. PDT.
Yeah,
gelatinase.
like, oh, for the TDT, you said you could email us about that, but
Biofilm.
biofilm, I'll email. You said you could do that.
You can, but put it right here if you have the time. These are two outstanding questions. What you learned from my BIOC17 and skills and stuff.
This one we already discussed it. That's
I
why
know.
I thought like it was like useless to write it down.
I would like you to actually think about it.
Okay.
And like if we have like 3 minutes, just show it to me whenever you're done. Those two. Very simple. Yeah.
So there's just the results. This is the platform results. And this is basically
We can separate and technically this will be the first.
well, this one, yeah, this one's going. This one's going.
It should be going down.
Yeah.
Update our purpose.
For the Bible sample?
Just for or actually technically I think it was both. But don't worry about this for now. I want to also make sure I answer Julia's question. But I will say check out purpose and other updates and see if you can potentially just reverse this order. So that way you see the 12
Like, since it's that it should be like just do like a fit here, like our version both like Bible comes again.
I would because isn't this one so you can do again? That's just I mean, no problem. I just want to answer a few other questions. I'll come to you next one. This one and Julia. I'm worried about time, so I'll answer yours.
Okay, so I'm just showing you my 11.
Okay, for week 9.
For week 9, we just did, yeah. So I basically formatted it like I would.
Okay.
Questions, my figure. And I think that the 3 plates, the Jazakumullahu test, and
Perfect. Okay. Good. Find out my 3. I'll come to you to show that.
So I'm still gonna add the numbers in there because I haven't done it yet, but this is what I had sort of planned out. I have to kind of put more depth into it a little bit, but this was what I was thinking for the table.
So I would recommend putting above each description a number.
Yeah, of course. So, but this is okay if I did it like this, almost just like explaining the growth on the sides, if I, or I feel like, because I'm explaining the plates here.
Yeah, I think it would be better this in the paragraph.
Okay, and so would you suggest if I put the numbers there, should I like say anything else, or just put like the numbers? Just the numbers and then explain it more to the figure caption and then a paragraph with it.
Yeah.
Okay, thank you so much.
No problem. I'll come to you shortly.
We also do a TDT graph for the photos.
Okay.
And then, so.
Can I see them, Jordan? Essentially, this is yes. And you want to extend it actually technically until and actually so then all the way to 0 for the TDT. So you're able to do this with the scale page. And essentially trying to do this either here or there, and just you'll see adjusting the scale for your PDT values, your D values, and how they compare for 50 and 60.
Okay. And then also the factors, which is 7, the results and as well as gelatinase test, or thinking and the glasses figure, Mannitol figure caption.
Any questions?
For the table, I know is it okay if I put all of the plate to the side? Because all of them are folded except epidermis and full 30. So I just put it for the B side. So sorry, the mask.
So this mask technically always should be consistent.
But I saw in the image that with their time 30 and colon and epidermis had like no growth for whatever reason. But when I put it on the table, I just put everything in one column. Like for all of them is 3, except in the last one. I put. Is that okay if I did that? Because I didn't want to have an extra 4 columns for like all of them. So I just kind of condensed all of 2 of them.
Can you put some minutes where you get that?
Oh, sure. I can show it to you right now, but you can just after as well. So this is like the observations for this week with the yeah. Wait, more like the answers, and then this is the biology, and I have to do the lysis element.
That's great. Thanks so much.
Big captions for the 3 plates.
I appreciate it.
And I decided to email you the
Today's observations for plates select differential. We've got 2 questions from Mr. Landis.
Okay. Number 6. Can you answer that as well?
Oh, it's in, it's the self experience, right?
As soon as you.
Okay.
So I just put in 1 column, all processes like that. So I put all of them 3, and then for the last one, for the 30 minutes,
That's
it was
okay.
3, but it was except equal line as epidermis was 0.
Really?
Yeah. Do you know the image was on the Turkish page? Okay. So I don't know if I'm like reading it wrong, but
So it could be possible that they would not pass.
we can just talk about the discussion.
So for those who are just
This Please validate if the topic right side. These groups where it's okay, that's
Right.
so we really can't actually promote that. So I'll okay, this I'll highlight. There's one down there. That's fine. No, but I was gonna find it.
And then for this one, I'll do what I said.
Let me see if I can mix that. Also, if that purpose, if I can alternate like the picture itself. But that should be okay. Go with the
because I see no growth because compared to like everything else, you can properly see like the yellow, like the.
Let me see the top one. The top left.
Oh, sorry.
Okay. Actually, that does make sense because we're at the last time, but at the same time,
Maybe I can read it like.
I think that's
For both of them. Thank you. And for the life factor, can we send that to you by on like Saturday because we just have a lot of assignments this week. Yeah, it's so hard. Like we have like 3 assignments due on Friday. Yeah. Please submit it like by Saturday because today I want to work on the echelon.
if you have shown me everything else for practice practice, then yes.
Okay, so like I said, we can send it to you on email.
Let me clarify for everyone else that is here. If you're done, reflective practice, 3 plates, the gelatinase test, the biofilms, and the reflective practice, you're done all 5 of those.
Yeah.
Then you can send me the lysozyme and TDT. Yeah, but technically you need to do the TDT for a microplate reader. So that can be done by Monday. But please do so.
We will.
Please send it to me by Monday.
We'll probably have it to you by Saturday or Sunday.
That would be amazing.
Because Friday, after Friday, I'm going to do all that.
That sounds great. No problem. Take care.
Well, that's why like you're doing full cycle, that's why you couldn't give personalized feedback.
Yes.
But then I think you said if you asked, yeah. So I was just wondering, but like I appreciate like it really, but like of course there's always so there was like a critical mistake I don't want to repeat it. If you want me to
Yes, that would be amazing. Hopefully you have in the comments, like in terms of the breakdown.
yeah, like I followed. Like that's how I read it. So what, like there must have been like some critical like a critical one is like, easy to like easy notes. So I just want to put that
I understand. So yes, you can email me in terms of referencing your grade per section, and based on what I've mentioned or what's shown on the outline, or actually just what we've talked about.
This after like or is this like where
This is for the upper level.
yes.
Yeah.
It's like what went wrong so I can just avoid it for the next.
That sounds good. If I can't because of timing today,
Oh no, that's not
yeah, I appreciate it. Yeah, let's see anything else. Is that okay? Yeah. Perfect. I appreciate it. So you know, that's a point. Okay, let's see for your case. Can I just see your input into that just to make sure I'm not yes.
For the result, like when you're looking at the picture, like when you're reading this in like Rule of 30, there's like a couple like I think mini like E. coli and epidermis. There's like a couple on like the A side where for like 25 minutes or 30 minutes. There's like two colonies or three colonies, or some of them have like 5. So would you rate that more like 0 or 0.5? That's it.
0.5.
If you get that like 2.
No, I would say 0. Or but put in brackets like only 2 columns.
Oh, so you could do like 0 and that's like okay, 3 columns.
Like a very brief observation. That's okay.
That's okay.
Like,
It's like in the table. Like
that's okay. But the cookie scale and in brackets like specific like details and details. Let's say it's like 4 colonies or 1 large colony or 2, 3 colonies. That's just not like you can actually put in the table. So hypothetically, you have brackets here. Let's say this is an anomaly. Let's say
like to explain the 1, 2, like what they represent.
yes.
Okay.
Yes. But if like there's some anomalies like this where you found like only 2 colonies or like a very large colony, you can describe that very quickly with scale, but it's like 0, 0.5.
Yeah. Thank you.
Also, the picture you sent, there's like 25 plates. Do you want us to?
You don't have to cut them up. You can just actually the entire
Like if I like say I put it on a side, this is the act rates, so I can refer to it easier. So would that be? Because like I heard you say like B is supposed to have this control, right? But some of them didn't. So that means that the A shouldn't be technically significant. We're like, because the control, if there's no exposure to UV, there should be growth. And if there's no growth, then something clearly went wrong. Like
you could have been wasn't much. So like this directly be exposure
exactly. So like something was wrong, right? So that plate should be. But like if I just say this plate, so do you want me to like put grids and put like 1, 2, 3, A, B, C, and then I just refer to A1?
But I will kind of already.
Okay.
I will get an idea, like based on what you described. If you get for example, equalize E30 on.
Okay, well it's already late. Okay.
They sure made a grid. That's it.
I didn't do it.
Okay, that's okay.
I had a 16 pages.
Yeah,
that's why I sent the pre-lab later.
That's okay. Thank you. Take care.
You wanted to see my notebook for this.
Oh yes, yes. Actually, it's cool. Some marks will actually ask you essentially for the reason. Oh, good. Is this be released about minus one?
Yeah, before, like I did it before.
So typically all this is from another lab, right? Like this.
Oh yeah, that's old.
Okay. So I was just.
Oh, because you're like, oh, this is the first time.
Yeah.
Okay, yeah.
So the reason I'm looking through patterns and stuff, so I'm just trying to see, okay, how.
It's like the biggest. But so like it depends on if you want to look further, like how much effort.
Yeah.
Okay.
And that's my environment.
Yeah.
Yeah. So for context, I said who is your group?
At the start, or
Wait. Seat. So look at it first.
you won, and then after you came, you looked around and you're like, actually it's seat. Before describing like these ones.
Let me give you. So there are 100% local quality, UV1. And so essentially the average of the two would be an average of lab performance and lab performance.
Because nobody is a.
That's what I was like modulating essentially. But for context,
Like if I was,
I will keep you at the D1 and then essentially account for your local quality. So you would essentially round off with just those two, by the way. So performance in the local will be at a D2. Or you'll be okay, you'll be at a 9. No.
for performance.
So the lab performance is a D1. I want you to assume you're at a 9. And then for lab notebook is a D. So the average is 2.
But they're two separate ones.
They're two separate ones.
Like,
But I do want to stress though, like either you have to put in more effort or you have to maintain effort to essentially keep up. Because otherwise, hypothetically, someone can move to C2 or if you're not putting pressing to the lab, let's say for this 3 points.
like I just try effort, such as like doing the graphs before coming here, because I know you gave time. Or because like I rejected in the discussion because like it's only 2 time points. You have to fit in this thing. So like even like a slight millimeter off, it would have been off.
I am actually so well like keep it up.
So like I've been trying to put in effort and like as well as doing like since she mentioned you could do this, but she didn't post it. So like I tried finding like an online one just to, like at least have it done to like understand.
But that is. But just for context, the police it's normally like down. If you're not the DNase, not the this is the biochem, it's just like shit. So maybe that's why DNase for the substrates. Yeah, like absorbance. But DNase for reaction. Yeah. So you're talking about the bulk lines. Yeah. Is that the page up 11?
No, that's not. That's that one's from like.
Okay.
This one was HS50.
Yes.
You did the CP3?
You know? So yeah, I know what you're talking about. Yeah. Do you have methods? I forgot if you use methods in that FLR. Yeah.
Like,
it's with Dr. Brent or another. Okay.
But like this one is so different because they completely like changed the way you write things.
I'm also recording, so I'm gonna tell that.
Take care of them.